Mendel's experiment was very perfect. He was successful because he kept all the data on record. Not only this, he was a great mathematician also. He used statistics very smartly and applied rather to conclude the results drawn by his experiment. These results gave rise to the laws of inheritance. Mendel performed his experiments in 3 stages. What were those stages? First of the stage was that he made sure that the plant which he had selected must be true breeding plant. By saying true breeding means the plant which we are studying should not be artificially born. It must produce good quality flower and good quality seeds. That is known as true breeding plant. It can undergo self-fertilization. So these are the characters of true breeding plant. The second important point regarding the Mendel's experiment, that he performed the process of cross-pollination of alternative form of a trait. Alternative forms were used by Mendel. For example, for tallness, there was alternative form of dwarfness. For yellow seed, to alternative form of green seed. For rounded seed, to alternative form wrinkled seeds. Likewise, there were 7 contrasting characters which were studied by Mendel. These are the 3 above mentioned characters which were studied by Mendel. Apart from these 3, there are 4 other characters also which Mendel studied while doing his experiment. These characters were flower color, next was pod color, next was seed shape and color, which I have mentioned. Seed color, seed shape, and pod shape, pod color, pod shape. Now we can count. There are the 7 contrasting characters. The reasons for Mendel's success was that these 7 contrasting characters did not have any intermediate form. Means from yellow and green there was no yellowish green flower. Yellow or wrinkled, there is no intermediate form. So they are absolutely visible from naked eyes. We can see them just by viewing them. By conducting the experiments, Mendel drew various 3 important rules. So let us see what were the experiments conducted by Mendel. The first experiment is known as monohybrid cross. In this cross, Mendel studied transmission of one contrasting character. And the one contrasting character is tallness to dwarfness. What he did initial stage, he took pure tall plant and ensured that they do not have any dwarfism genes present in it, and another is pure dwarf plant, and first of all obtained gametes out of it. It can produce only one type of gamete, it can also produce one type of gamete. When these two gametes meets, again there is hybrid tall. Now let us once go back to the basics, that why it is tall, because of this allele capital T. Whenever this capital T is present, the plant is bound to be tall, because this is a dominant gene. This is F1 generation. Now Mendel cross-pollinated the same flowers to obtain the F2 generation. It is seen by drawing this chi-square board or checkerboard. Here we write the genotype of male and female. So when we cross-pollinate, we have to write, because it is the same flower, so same genotype has to be written over here. Capital T, small t, capital T, small t. Now we will cross in the same way. Here one should remember that the gene for dominance or the allele for dominance should be written prior to the recessive. So it will be like this. First we will write capital T, then we will write small t. Here we will write capital T, then we will write this small t. And if you will cross small t with small t, it is bound to come small t and small t. So the genotype ratio in F2 generation is capital T, there is one plant, two plants with the genotype small t, so it is one capital T, two capital T small t, and here only one capital small t and small t. So it is mentioned 1:2:1. But since this is hybrid tall, so the phenotypic ratio is mentioned as 3:1, because 3 plants are tall and 1 is dwarf. So if we conclude here, the genotype ratio in F1 generation, it is 1:2:1, and the phenotype ratio is 3:1. Coming to the next, dihybrid cross. Mendel studied two contrasting characters during this cross, that is seed color and seed shape. Seed color for dominance was yellow, so it is written as capital Y, capital Y. Its contrasting form was green, so it was written as a small y, small y. Seed shape dominant is rounded form, written as capital R, capital R, and dwarf the recessive seeds are mentioned as small r, small r. So these are recessive traits and these are dominant traits. If we write the cross type here, so it should be written like this: capital Y, small r, capital R. Similarly, small y, small r. Now we should remember that it is for yellow seed and rounded form. It is for green seed and wrinkled form. When we will obtain gametes out of these seeds, only one of them will come because of meiosis. Here again small y, small r. If we see the hybrid F1 generation, the plants obtained here, they are of this genotype: capital Y, capital R, small r. This is a hybrid seed with yellow color and rounded form. Now we should remember why it is rounded. It is rounded because of presence of capital R allele, and why it is yellow? It is because of presence of capital Y allele. Now Mendel self-pollinated F1 plant to obtain the F2 generation. The results were very interesting in F2 generation.